Here. Welcome to the next installment of Alberta Innovates BioSolutions Impact Innovation. I've recognized many familiar faces in the foyer, but if you're not too shy, can you indicate whether you're here for the first time? Anybody here for the first time? You're very welcome. Thank you very much. That's great to see you. A small round of applause, very appropriate. Indeed, this is Alberta Innovates BioSolutions public event. We're looking forward to just a wide range of fascinating topics. I'd certainly like to acknowledge our board chair, Mr. Art Froelich, who gets a $25 seat at the very front here. Mr. Art Froelich, very good. And you'll be seeing a couple of our other board members later on, but I'm not going to spoil that fun, although I do know you have an agenda. Let's do some housekeeping. If you have phones, if you could sort of dial them back to silent, that would be fantastic. Washrooms are outside in the foyer and hard left or right if you wish. Don't be alarmed. There might be a bit more technology this particular year. In addition to what's going on here in the hall, we're live streaming this event across Alberta, Canada, the world. So we've given you some options. You can do things here in Ziedler Hall. You can be streaming, looking at this event from wherever you are. If you're in the hall and live streaming, I think you have to reevaluate your relationship with technology. But let's see how that goes. So what does Alberta Innovates BioSolutions do? We're given the responsibility from the government of Alberta to make investments in agriculture, in food, in forestry, in the province of Alberta. Those of you who know me know that I never miss an opportunity to remind people, based on the most recent stats, what are the sales of those three sectors here in the province of Alberta, of agriculture, of food, of forestry? Shout out a number in the billions. I heard a number, shout it louder. 25, actually, most recent data now, $27 billion worth of sales. So these are important industries to our province. They are renewable industries, and Alberta Innovates BioSolutions is very focused. We know that's not an entitlement, so we're very focused on thinking about how do we keep those sectors profitable? How do we make sure that they are sustainable in all the ways that we would anticipate? How do we do that? We make investments in sustainable production, in innovation in food, bioindustrial refining, prions, ecosystem services, and you're going to hear a little piece of each one of those during this morning. It gives me great pleasure to introduce someone who is the author of at least 12 books, a member of the Order of Canada, a well-known Canadian broadcaster, and I've always wanted to use this word in an introduction, and a raconteur. Ladies and gentlemen, Jay Ingram.
Morning, everybody. This is going to be a very interesting, stimulating, I think quite fascinating morning. You know, it's unfortunate that the rest of Canada thinks of Alberta as 7 months of, well, actually be like 8.5 months of winter. Two hockey teams that never make the playoffs, and oil and gas. And as Stan indicated, food, agriculture, forestry are huge industries, not just here, but in fact have an impact across the country and around the world. And AI Bio, you can't have those kinds of sales and that kind of industry unless you have a robust research program underlying it, and that's what AI Bio does. So this morning, we're going to have 6 presentations, 5 from researchers active in quite different areas, although in a second I'll try and outline how I think they relate. And another one from that interesting combination of researcher and entrepreneur. So sustainability, something that the oil patch continuously struggles with, I think is one of the watchwords in AI Bio. And you will see today that sustainability in one way or another underlies all this research. So for instance, you have the automobile industry becoming more sustainable. You have beef production becoming more sustainable. Those both you'll hear about more. The other thing is there are interesting and I think unexpected connections between all of these areas of research. So for instance, the idea of reducing carbon footprint, that ties together John Basarb's presentation on sustainable beef and Will Harney's on the use of biomass in automobile manufacture. If you think about the health of Alberta's human population and wildlife populations. We'll hear from Dr. Valerie Sim on prions and Dr. Raylene Reimer on combating obesity. And Scott Nielsen will extend both those ideas to the health, not just of humans and animals, but virtually every living thing in the province. So let's begin with health. And I'm going to give you some startling statistics. In the 25 or 26 years between 1985 and 2011, adult obesity in Canada tripled from 6% of the population to 18%. And if you extend that trend into the future, and I don't think there's any real reason not to, you come up with a stat that I call 5 and 5. That is, in 5 years from now, 2019, there will be, in 5 provinces in this country, more overweight or obese adults than normal weight adults. And it's not just an adult problem, and this, I think, should be an even greater concern. 12% of Canadian children and youth are now obese, 19% are overweight. And obesity brings multiple health complications, and it's a problem that I think the problem is underappreciated in that its roots are much more complex than people will give it credit for. The other day there was a piece in the National Post about British researchers claiming that their studies of fiber in diet would allow them to combat the problem of obesity. And what the Post either didn't know or didn't choose to say is that in fact advanced research on this very topic goes on here in the province of Alberta. And it's being done by Dr. Raylene Reimer. She's a professor of kinesiology and medicine at the University of Calgary. Raylene.
Good morning. Thank you, Jay, for that introduction, which I think really nicely sets the stage for my presentation today. And you can see that the original title I came up with was Adding Value to Food and Life. But as you'll see in the program, I think Jay came up with a much more captivating title, the full meaning of which I think you will understand by the end of my talk. So what I'd like to begin with is a statement which really underlies the motivation for the research that I conduct. And this statement is that the longer obesity persists, the more it changes your biology. So really, what does that mean? And we have some excellent understanding of obesity and its causes and what caused the maintenance of obesity from appetite regulation. So we know in the body there is this very complex yet integrated system of signals that travel from our digestive system to the brain to indicate to us whether we feel full, do we feel hungry, and to really monitor and modify our food intake. In that system, we have factors which I like to think of as the red light signals. These are the signals that travel to our brain and they tell us we're full. We can stop eating right now. And just one of those signals, amongst many, is called PYY. On the other side of the appetite and eating behavior continuum are those signals which I like to think of as the green light signals. And these give the body the go-ahead to eat. So they're the hunger drive within us. And for centuries, these signals have worked in a very nice manner to, in essence, allow adults to maintain a very stable body weight. But within the past generation, we know that these signals are very susceptible to the environment in which we live, and in essence, are susceptible to what we call the post-dieting syndrome. So what is the post-dieting syndrome? Well, we have some very good understanding of what this means and why individuals actually struggle with overweight and weight maintenance is in some research studies that were conducted in individuals who had been overweight or obese for many, many years. And in the study, they were assisted in losing 10% of their body weight. So if an individual started at 240 pounds, they lost 24 pounds in the study. And then they were asked to maintain this new lower body weight for the year. And at the end of this year, they did a whole battery of tests on these individuals. And this is what they found. First of all, in terms of appetite regulation, what they saw a full year out from the weight loss was that ghrelin, this hunger signal, was 20% higher. PYY, that factor that tells you we're full, we're satisfied, was actually reduced 20% lower than it was at the beginning of the study before they lost the weight. And in essence, what the body was telling these individuals, or their brain, is we're starving. The body, of course, was not starving, but those were the cues that were going to the brain. And it was driving the individual to regain or defend back to that higher body weight that they started at. It doesn't end just with appetite regulation. They also have done studies where they can see that there are changes in the number of calories that the body burns post-weight loss. So again, after having individuals lose the weight, they took a muscle biopsy and they looked at how much energy is being burned in that muscle tissue in daily activity and exercises. So going out for a brisk walk, doing the yard work. And what they saw was that the muscle was burning 20 to 25% less energy than it had prior to their weight loss. Again, this biology was driving the individuals to gain back the weight that they had lost. And it doesn't stop there. The brain was also altered after the weight loss. And so this burger, of course, well, maybe not at 9:30 in the morning, doesn't look too rewarding or appetizing, but it should. And so the reward centers in the brain after the weight loss would see this cheeseburger as even more rewarding and drive the individual to consume more of these types of food, and the reward center in the brain would be highly activated. And so it makes it very difficult for an individual to exercise restraint. And so I think what this collection of research study tells us, it's powerful knowledge for why we struggle with weight loss, and more importantly, maintaining that new lower body weight. So for me as a researcher, the question then becomes, if we know this biology is being changed by the persistence of overweight or obesity, so the longer we carry the weight, is there a way to counteract this new biology that's been created by the persistent obesity? And of course, my training and expertise is in the nutritional sciences, and so I come at this question from a nutrition standpoint. And one dietary change that we know is particularly powerful in this regard is a prebiotic fiber intervention. And so prebiotic fiber is a particular type of dietary fiber found naturally in garlic, leeks, onions, asparagus, some in wheat and bananas. And if you think about our typical North American diet, we don't eat very much of this type of fiber. It's only about 2 grams per day. But if you eat sufficient amounts of prebiotic fiber, we know there's a whole host of benefits to the person consuming them. So it increases GLP-1 and PYY, these signals that travel to the brain and tell us we're full. It decreases the hunger signal, ghrelin. It can reduce voluntary food intake by individuals and reduce the subjective feelings of hunger that we have. Another thing it can do is improve the way in which the body utilizes glucose, which is important for prevention of type 2 diabetes and other metabolic disorders. And perhaps the most surprising of those is this statement that it improves the profile of the bacteria that live in our intestine. So why might that be important, of shifting the bacteria that live within our intestine? And this is an area of intense interest in the research in understanding the link between the bacteria in our intestine and the involvement in a variety of different diseases. And obesity was the first disease in which this shift in bacteria was actually characterized. And we know that if you look at an obese mouse versus a lean one, and we know the same thing is seen in a lean versus an obese adult or human, that there is a shift in the bacteria in the intestine. And this can actually be a factor that contributes to obesity, but also can modify or allow for the persistence of that obesity to take place in the body. And one particular type of bacteria in the intestine is bifidobacteria. And these are typically considered healthy. We see them in probiotic yogurts and whatnot. And these are very much reduced in the obese situation. And for me, the good news here is the more we learn about this gut microbiota or all these bacteria in the intestine is we can actually manipulate them with diet, and we can shift them back into what we would consider a healthy profile. So I would just briefly like to describe for you some of the clinical trial work we do in terms of dietary fiber with the management of body weight. And in this study, we used a prebiotic fiber supplement and asked the individuals who were overweight and obese to take it for 3 months. Now, what you can see here, these white bars are the control or the placebo group. They simply, in essence, got a sugar pill throughout the 3 months. And what you see here is over the 3 months, they actually gained a little bit of weight, so approximately 0.5 kilogram or 1 pound. And what we know is that the typical adult will gain approximately 1 kilogram or just over 2 pounds every year in adult life. So this is what we saw in the placebo group was actually very typical of what happens to us in adulthood. And a little bit of that, about half of that was fat mass. Then we turn to the black bars, which are the subjects in the study who received the prebiotic fiber. And what we can see is not only did that prebiotic fiber stop this typical weight gain that we all seem to experience in adulthood, but it actually caused them to lose approximately 1 kilogram, and the vast majority of that came from fat mass. And along with that, we see that it then decreased ghrelin, the hunger signal, and increased PYY. But certainly prebiotic fiber is not the only type of fiber we need. It's not the only one that can have benefit. And so with funding from Alberta Innovates Biosolutions, we looked at another unique type of fiber source, and that's from the dried yellow pea. And again, in this study, we had overweight and obese individuals consume an additional 15 grams of the pea fiber over the course of 3 months. And at the end of the study, we wanted to assess their appetite and food intake. So we gave them a pizza buffet. So all the cheese pizza you could want, and simply instructed them to, when you feel satisfied, stop eating, and then we do detailed calculations to see how many calories or how much energy they actually consumed. And we do that at the beginning of the study, at baseline, and at the end of the study. And those individuals who had been put into the control group actually ate more pizza at the end of the study than they did at the beginning of the study. So we seem to, unfortunately, drive up appetite a little bit in the control group over time. And those that consumed the yellow pea fiber actually decreased the amount of pizza they ate at the end of the study by 131 calories. And together with that, we saw the typical increase in body weight in the control group and decrease in body weight and decrease in body fat in those who consumed the pea fiber. Now, the way in which we actually incorporated the yellow pea fiber for the study was into a cookie biscuit. And this was developed with colleagues at the Food Processing Development Center in Leduc. And we, in essence, created this ginger snap type cookie. But that's not the only place that this yellow pea fiber could be added, and there's many, many other applications in terms of food. You could put it into soup, into muffins, many different applications. And so we know that Alberta crops really represent for us a rich source of potential healthy food ingredients that can be added into foods, which will bring value to the health of Canadians and increase the economy as well. So in conclusion, I would just like to reiterate that we now have some very meaningful insight into what the persistence or the long-term consequences are of carrying excess body weight. And obesity really truly does create within the body a new biology. And so we're looking for ways to combat that. And of course, dietary fiber is literally one out of many different targets that can be used in this sense, and it has numerous benefits that we've highlighted. And finally, there's real value in adding these high-quality ingredients that have health impact into the food supply. And so with that, I would just like to put up an acknowledgement of the many, many different trainees who have helped generate this kind of research, and for the funding bodies. Thank you.
That's good. I should add that I think that many of you might have questions for the speakers. We're going to allow 10 minutes just before coffee. We'll bring back up the first 3 speakers and you can ask questions. And questions like, where do I get that yellow pea pizza? And if none of you ask that question, I certainly will. And I said in my introduction that one shouldn't think of AI Bio as serving only Alberta, not just nationally, but globally. And when you consider the growth, the dramatic growth of the world population, things like combating obesity are not just a local issue, but a global issue. And so is raising animals for food. Now, traditionally, people worried about the growth of the world population, worried that that's much more intensive than just growing crops. But that's in the traditional methods. And there's no signs, of course, that the growing world population is losing interest in eating meat. So you have an issue. How do you make meat production more sustainable? Well, I also mentioned connections in my introduction. And in this particular situation, you can take the booming science of genomics and apply it to meat production. That's what John Basarb is doing. He is a research scientist with Alberta Agriculture and Rural Development, adjunct professor both at the University of Alberta and the University of Manitoba. John.
Well, thank you, Jay. Great to be here. And before I start, I'd just like to acknowledge right up front some of the research teams and funders that are involved instead of at the end. The topic, as Jay said, is I'm going to be talking about sustainable beef production, but from a technology point of view, I'd like to just acknowledge and go through some of the advances that have been made. I put this up here just to sort of get your attention and to show you the whole area of sustainability in beef production is really forefront in the food service industry and in the consumer's mind. And in this particular case, McDonald's has stated publicly in a release in January that in 2016 they will start sourcing sustainable beef production. Whatever that means, we're not sure. And that's interesting. And the reason I say that is, and the beauty of some of these large food service companies is they are aligning themselves with some pretty important individuals. In other words, McDonald's and Walmart are aligning themselves with what we call the Global Roundtable on Sustainable Beef Production. Many, many stakeholders are involved in here. And really what they are saying is that, at the general sense, they're establishing principles of what this means. And it really means that it is a balance between the resources that the world has in the air quality and water quality and the development of individuals and communities. Rural development has to be in there. It is also about looking at having food security, safe food, affordable food, and food that has good quality. That needs to be balanced with animal welfare issues, codes of practice, and of course, the health of animals, which is so important. The topic that I'm going to be covering a little bit in more detail is this whole area around progress. In order to be sustainable, you have to have progress, scientific progress in technologies, but also in terms of organizational management and how we look at the structure of these various systems. Well, I like to go to this really nice piece of work that was developed by that was researched by Judith Kapper in the U.S. It's about the sustainability of North American production systems over the past 30 years, and essentially science and technology has made some of these changes. So if I say the same amount of beef now requires 88% of the water, 81% of the feed, 70% of the animals, 67% of the land. And all of that results in about a 16 to 20% decrease in the carbon footprint of beef production. And what I'd like to say is that all of these changes have come about due to science and technology, advances in life science and technology in the area of nutritional management, health management, feed additives, the area of crossbreeding, and what I would call traditional breeding practices. Maybe I'll call that Breeding 101. So what's the problem? Seems like we're doing okay, so what's the problem? Well, the big problem is that we now have to do this faster, and we have to do this with more attention being paid to the social well-being, the environmental friendliness of a practice, and of course, it all needs to be economically sustainable. If it's not economically viable, then it's very unlikely that these types of things will go. Well, there's a couple solutions. There's more than a couple, but I'm going to dwell on just two of them. One is that we could continue on in what we're doing in terms of improving system production management, and it will make some pretty good improvements. Here's an example that I like to bring because it's a homegrown example. It's called swath grazing. A lot of work that we do at Lacombe Research Station is involved here, but essentially what it does is it takes advantage of our knowledge of the system. It says, you're a cow, go out and get your food. Right? Instead of us sort of collecting it off the field, spending energy to bring it into a feeding place, and then hauling the manure back onto the field. So it is a very sustainable type of a system. You can see the numbers at the bottom in terms of fuel savings, cost of feed savings, and the reduction in land use. The other technology, of course, is one that I really enjoy working in, and that's the area of genomic selection. And it's what I would call breeding 2.0. So that's just sort of to put it into perspective. Essentially what we're taking is old techniques that we've known for many, many years in traditional animal breeding and applying genomics to it. And really, in this particular technique, what we need is a really large reference population. And this is where funders like ALMA, Alberta Meat and Livestock Agency, and of course, Alberta Innovates Biosolutions comes into play. We developed this large reference population where we have animals that have really detailed phenotypes, characteristics, and really detailed genotypes. Let us say 50,000 bits of genetic information that we look at. And those bits of information, we're going to call them SNPs. So remember that. I'm going to tell you what that means, but I'm just going to say those basic units of inheritance are called SNPs, and we'll be looking at those very carefully. So what is really required here is that we then have to use this reference population to train our prediction equations. And we do this in this example. It's just a small example. You see that AT, that represents just one of those SNPs on a chromosomal pair. And you notice below we've got some various alleles. We've got AA, AT, and TT. And you'll see that as we change those alleles, it's related to different levels, in this case, of growth. So AA is associated with 0 grams increased growth per day, whereas TT is represented by 40. And essentially what we'll do is simultaneously fit all 50,000 SNPs into a prediction equation, and then use that prediction equation to create a score. You know, how well did you do at school, kind of a deal. And then we'll take hair samples or blood samples or any kind of tissue sample from what we'd call our selection candidates and use the molecular breeding values to decide on how we deal with them. Well, let's look at, here is our population. I'm proud of this. This took probably over 5-6 years to create. hate to say, but about $3 to $4 million. So these types of databases just do not get created. As you can see, we've got about 9,000 cattle with our favorite trait, which is called residual feed intake. And we've got the same 9,000 animals that have detailed genotype, 50,000 bits of genetic material. This scatter graph here, each dot represents an animal. So on there's over 9,000 animals, and you can see they vary quite nicely in terms of their summer efficient. So those animals that are below 0 are animals that eat less but produce the same amount. And those animals above the 0 line on the y-axis are those animals that eat more but produce the same amount. A colleague of mine, Erasmus Okine, he essentially said, those animals are eating for fun, right? They're really not doing anything with them. But I know I've gotten in trouble several times, Erasmus, for saying that, so that's just between you and I. So let's just take a look. From that large database, what can we do? Well, the very first thing we can do is we can create what we call traditional breeding values, estimated breeding values, okay? And we could use those, and we can select animals that have good estimated breeding values for feed efficiency. And when we do that, we end up, and we take those bulls, and we mate them to a bunch of cows, and we end up with efficient progeny. We can do that and use it in the feedlot, and we can save about $10 to $15 in feed relative to an inefficient sire. You'll notice, and I'm going to bring this back later, is that the accuracy of these estimated breeding values is pretty good, 55 to 65%. You might not know much about that, but just note that for now. The other way of looking at this is one around sustainability. If I create 6,500 efficient feeder cattle and take them through the feedlot or take them through a year in a cow herd, I will save 475 tons of barley. That's that pile there. I measured it. It not only will save 475 tons of barley, but there will be a decrease in land use. And there will be a decrease in the carbon footprint. In other words, we're trying to improve the sustainability of beef. However, it took us a long time to do that and a lot of money. It took us 5 or 6 years just to be able to get those estimated breeding values and to create that database. So then the other thing we can do is, of course, molecular breeding values. We can take that same database and then create those prediction equations and just take a blood sample from an animal and say, how did you do? How good are you? This particular overhead just shows some traits we have, our residual feed intake, which is our favorite feed efficiency trait, and then a couple meat quality traits. But you'll notice right off the bat, the accuracy of these particular molecular breeding values are not as good. So it sort of decreases the progress that we can make. But exciting things are happening right now in the area of livestock genomics, in the area of sequencing. The cost of sequencing has dropped dramatically. And so the next level that we are going through, the world is going through right now, is in the whole area of using all of the genetic material from very, very popular bulls or individuals within each breed. And I'm going to show you how that works right now. Now, this particularly case is where within the Canadian Cattle Genomics Project, animals, very influential animals across the world in many breeds. You'll see the breeds down the side, but it's not that important. There's 10 breeds or crossbreds that are used here. And they were fully sequenced, and they're sequenced at about $10,000 a piece. And the very first thing you'll see is that there's about 12 to 16 million bits of genetic variability that are related to some characteristic of the animal. That's tremendous. Also in that particular case, there's about 1 to 2 million more bits of genetic variability called indels. These are insertions and deletions. You'll see sort of a little graph on the side that shows you what an indel type variability is. And these are naturally occurring, by the way. We didn't put them there. They're naturally occurring. Now, the exciting thing is when you look across all these breeds of cattle, there are 30 million bits of variability. 80% of those bits of variability have never been defined. This gives us tremendous opportunity for improving the accuracy. I'm going to show you, just in my very last slide before concluding, how this occurs. Essentially what we do in the very first row there, we have a low-density genotype. In other words, a 50,000 SNP genotype that we have on an animal for around $80. We can have lower density genotypes for less money. And essentially you'll see right at the top there is that we have a pretty, not a very dense genotype on that chromosome, right? Every so often we end up with a genotype. The trick is, how can we fill in all those dots? How can we fill in the blanks? And that is through this process called imputation. It's a quantitative geneticist word. They love to make these words up. But essentially what it is we're extrapolating from the full sequence data to what this actually means. Let me show you. In the middle, we have our whole DNA sequence data. It costs us $10,000 an animal to get it. 300 animals, that's $3 million. This is tremendously valuable information. And let's go a little bit further. Just look at that segment that we're looking at. In these animals, these influential animals, we see that the most common sequence for that particular location is CGAGA. And so that's your answer, right? It'd be lovely if we had a course like that where we could actually put the answer. So essentially what we get is we get the actual sequence of a $40 to $80 test coming from $3 million worth of information and even more. And this will have tremendous improvements on improving the accuracy of our breeding values. Okay, in conclusion, here are some cows. No, in conclusion, the beef industry will continue to get better at sustainability. It will continue to use technology in all sorts of areas. Genomic technology will be one of those major technologies that has the potential to double the rate of genetic progress, particularly for difficult traits like feed efficiency, carcass and meat quality characteristics, and a low methane phenotype as an example, all towards getting a more sustainable beef animal, or what I call the climate-friendly beef cow. Thank you.
Thanks, Jeff.
If we could interrupt for a moment. We have about 10 or 15 people still looking for seats. If you want to tighten things up or indicate where a seat is available, we probably have a few open seats. We know that we're not like Air Canada. We have not overbooked. So just find your way in. Just help out the people that don't quite have seats yet, and we'll carry on. Sorry about that.
All right. All I want to know is who's ever been to a day of science talks where the first two each have a slide of a cheeseburger. We didn't plan that. You know, with reference to what John was talking about, I suspect it's a big relief to Albertans that we can actually be thinking about a healthy beef industry when 10 years ago we might have been worried if there was going to be a beef industry in this province at all. But that's, of course, because in May 2003, Stephanie Shub, who's also an AI bio researcher, was alone on a Victoria Day weekend at the National Microbiology Lab in Winnipeg, and she was the first person in the world to see that we had a case of mad cow disease. And as I'm sure all of you know, it was a terrible blow to the beef industry, which is in some ways, I think, still recovering. Now, the silver lining to that very dark cloud was the creation here in Edmonton of an internationally recognized prion research lab, prions being the causative agents of mad cow disease and many others. Dr. Valerie Sim is one of the AI bio-funded researchers at that center, the Center for Prions and Protein Folding Diseases here at the University of Alberta. Valerie's also a practicing neurologist, so having one foot in the clinic dealing with human neurodegenerative disease, including prion diseases, and one in the lab, I think gives her a privileged view of prion science. Please welcome Dr. Valerie Sim.
Right, thank you very much. And I too would like to echo my pleasure in being here to speak to you today. I think it's fascinating, the theme in our slides, because I too didn't plan this slide knowing my last speaker's last slide, but this is a bit of my theme as well. So today I'd like to tell you about my passion, my research in prion disease, and how a twisted protein can kill your brain. So what are prion diseases? Well, the technical name for them is transmissible spongiform encephalopathy, which is why we say prions, because that's a bit of a mouthful. And this includes a number of different diseases. In humans, it can cause the following diseases. It affects sheep in a disease called scrapie. Of course, as you heard, it affects cattle in the disease we know as mad cow disease. And maybe some of you have heard of chronic wasting disease. It affects deer, elk, and moose. And I'll talk a bit more about that disease later. And fortunately for me as a researcher, it also affects rodents, because this is the mainstay of some of the way we do our research. So in humans, this disease is always fatal. Early on, people come with difficulty with coordination, some trouble thinking, and as they progress, they stop being able to talk, they stop being able to move until they die. And any idea how long this disease takes, the average time to death after symptoms starts?
1 year.
4 months. For the most common form of this disease, 4 months. So this is a very fast disease. It's what we call a rapidly progressive dementia. So what causes this? As Jay mentioned, these are caused by prions. Well, what is a prion? The interesting thing about these diseases, they can happen spontaneously. In fact, 90% of cases are spontaneous, not related to eating contaminated food, anything like that. But you can also be infected with this from surgery or blood transfusions. And not only that, you can inherit this disease. So a very unusual type of disease. And the infectious agent itself is really difficult to kill, unlike typical viruses or bacteria. So what makes prions unique? Well, if you think of a virus normally will infect a cell, inject its DNA or RNA, replicate that, make new viruses, and go on with its life cycle. Well, a prion does the same thing. It infects a cell, replicates itself, moves on, but it's only protein. So how on earth does that work? That's the fascinating bit about prions. Well, in order to explain what we know, I have to introduce you to a protein that I spend a lot of time with. It's called the prion protein. Now, protein isn't just what you add to milkshakes at the store. Proteins are actually molecules that are made inside our body and do many, many different functions. We couldn't survive without proteins. This particular protein lives on the surface of our cells. It lives on cells in our brain, in our heart, in fact, all over our body. And you can see we know a lot about this protein. This is actually what it looks like. This is its shape. You can see I've got all these labels on it, and it's not actually pink and yellow, but the shape is correct. And the shape is really important because proteins function based on shape. So the problem in prion disease is this protein changes shape. Now you notice I don't have a bunch of diagrams on labels because it doesn't actually turn into a bunch of pink triangles. But we don't actually know the shape, but it does change. And the problem is once it changes shape and it sidles up to a normal prion protein, it makes it change shape. And then this process continues and you get a buildup of these misfolded proteins. So shape is really important. And that's what I'd like to talk about in sort of the first project that my lab's been involved with. Some of you may have seen this article, it was last year in March, which talked a bit about our research and the importance of shape, and not just shape, but shape shifting. So if you imagine the prion protein doesn't just form pink triangles, but it can form any of these shapes. All right, squares, diamonds, any array of shapes, or what I would call a cloud of different shapes. Now, one particular species can form a cloud that has red squares and blue diamonds and green triangles. A different species, that prion protein, might be able to form the other shapes. So if you take one pink square and square, diamond, and triangle from species 1 and try to infect species 2, it's not going to work. The shapes don't match. Aha, but what happens if it encounters another species whose cloud overlaps species 1. Well, now I think you can see you can pass from species 1 to 3. Well, let's introduce another species, and I think you see how it can progress from one species through to the final species. Now, this isn't necessarily only the case for between different species, but it can actually happen within species as well. And that's sort of what we found, and I don't seem to be able to advance the slide right now, so I'll let you figure that out. But this can happen between, say, a different animal, such as in chronic wasting disease, deer or elk or caribou, and could that then transfer into humans by going through an intermediate host? And this is a very serious concern we have right now. So another project that we're looking at, so in addition to just shape, is if you imagine you have all these red triangles or red shapes, how on earth do you get from that to actually causing a person to die in 4 months? You know, what's happening there? And we know that these proteins, these shapes accumulate in the brain, and then the brain itself develops holes, full of holes. In fact, that's why the disease is called spongiform encephalopathy, because it starts to look like a sponge. So something we're interested in looking at is the brain itself and what happens during disease. To study this, do I have hope? No? Okay, well, I will pretend I'm a brain with holes in it. I should have brought my fiddle, I knew it. Anyway. So basically, you develop holes in the brain, but it's a bit difficult to say, look at the brain as it is without some help. I saw a brief glimpse there. Oh, so, okay, I'll quickly run with this as it's going. There it is. All right, I'll go back one slide. This particular approach that I have, we call prion disease in a dish. And basically, we take an animal, a mouse, and we use its brain, sort of the back part of the brain. We slice it up, and we actually plate it in a dish. This is starting to sound like food again, but we don't actually eat this. Here we can actually grow the piece of brain. For weeks and weeks and weeks, we can infect it with the prions, this infectious material, and it will actually, we can monitor it over time and see if it develops disease. So when we look at this, this is the actual brain slice now that's been growing in a dish for 35 up to 49 days. And we've colored it, the different cells. So a lot of the neurons are stained white, and another special type of neuron is stained green. And they're very healthy looking. You see, over time, everybody's doing well. And this is an uninfected slice. Well, compare that now to an infected slice. And I hope you can appreciate that with time, we lose those neurons, those nerve cells. The white signal is almost completely gone. Now, if we look more closely at an area in the infected slice, the green area, we can look at one of these green cells. Oh, it's freezing again. Technology's failing. Yeah, it's all right. It's the only problem with images, because I don't know if I can pretend that I'm a big neuron. But on these big green neurons, there's a bunch of little spines, so these little processes, because in order for a neuron to communicate with other neurons, you imagine my hand and my fingers, it has to talk to my other hand and fingers. And these fingers are important for communication. These fingers are important for how neurons talk to one another. And what we see in the animal is those fingers are lost as the disease progresses. And what we found in our slices is that when we watched for those signs as well, we also saw that those fingers were lost. There they are. Quick. All right. So these are the little fingers that I was talking about. And then in disease, they vanish, they disappear. And the good thing about this is it really means that this model, it's not just something growing in a dish that can sort of make prions, but this is actually replicating some of the disease. And it does it faster than we see in the animal. I'm talking about 40 to 50 days here. In some animal models, we can be talking for hundreds of days, potentially years, to see the same things. Plus, it's in a dish, so it's a lot easier to study and to look at. So what we have funding from AI Bio is to apply this technique to a bunch of different situations. So specifically, we're interested in, as much as we care for the mice, we're interested in other species and other strains. So we've actually started looking at chronic wasting disease. And here's just a couple pictures. This is actually after 70 days of culture. And using low dosages of the prions to try to infect the slices, they actually stay quite healthy, as you can see by all the white staining. But if we use a higher dose, by 70 days, they're quite unhealthy and they're starting to also go through the disease process. The little bit of red you see as well is a sign of active dying cells. We're also applying this to other parts of the brain. As I showed you, we're just using the back part of the brain right now. Well, all of the brain can be affected by prions, and different shapes of prions affect different parts of the brain. So we want to study that. So we've started culturing and infecting other areas of the brain. And this is sort of an area sliced like this through the brain. And again, showing nice healthy cells over time. And we've been able to infect these as well. Finally, it also gives us a place to test therapies a lot faster than in an animal or combinations of therapies. And so if I look at a slice here that's been infected, that's not treated, that's looking unhappy, as my technician would say, once we treat it, we start to see a bit of rescue. So some of those neurons are surviving now. And so this gives us a way to screen and hopefully advance treatment ideas for these diseases. So you may say, well, why? Why should we worry about this? You know, is mad cow, is that around anymore? Is it a problem? Of course, as a clinician, I care a lot about the humans that still get this disease. But I think you should also be aware of chronic wasting disease. Because in Alberta, it first appeared in a farm in 2002 in white-tailed deer. By 2005, it was in wild mule deer. By 2012, it was in wild moose. And if you look at a map of how this is progressing, how this is spreading, in 2007, this sort of gives you a North American view of chronic wasting disease. I'd like you to pay attention to the gray squares, the light gray squares. That's all of the chronic wasting disease in wild, free-ranging populations. And I'll show you over time what's been happening. And this map now is current as of April 2004. And I'd like you to note in particular Alberta. So this is a growing and rising concern. We don't know all of the implications yet to human health, to tourism, to the industry, but it's certainly not going away. So with that, I would like to thank all my collaborators, my funding agencies, and for your attention.
She deserves a lot of applause for coping
Yes.
with the technology glitch. I've seen another great example of that. We're just getting ready for the Q&A, so if anybody has questions, we have people with microphones on both sides. Because we're live streaming this, I'd appreciate it if, are we going to pass the mics into the audience? Okay. So if you have a question, just raise your hand. Somebody will bring you a microphone. I had to introduce Chris Hadfield at a Calgary Teachers Convention a couple of months ago, and he was talking about how difficult it was to get into the shuttle and how awkward. You've got this huge suit, you've got to walk across a catwalk, it's 200 feet above the ground. He's scared of heights. And then he is. And then when you get into the shuttle, of course, it's pointed nose up, so you have to clamber around. And he said, but the amazing thing, he's got pictures of all this, amazing thing is the vibration and the incredible noise. And he starts a video, and you see the shuttle there and the flame coming out and it's shaking, it starts to lift off, and it's completely silent. And so he stands and watches the video for about 6 seconds and then says, you know, I've been there and I remember it being a lot louder than that. So I think Val did a fabulous job of dealing with that. And we have a question from the audience right here. Thank you.
This is a
Go
question
ahead.
for Dr. Reimer. So when you mentioned the healthier sources of fiber, like the pea fiber, and then they're put into a cookie, was there any aspect of the study that was done on, the presentation of the food? So then you get used to eating cookies, and if you're eating healthier cookies, that's okay. But what about the future?
Absolutely. So the form in which you add these ingredients can certainly influence what the subjects do during the study. We tried, within the context of the research, we try and control for that by also giving a cookie to those individuals that were in the control group. So they ate cookies as well, just like the ones that got the pea fiber enriched cookies did. But I think We're not necessarily trying to encourage individuals to eat more cookies every day. That's probably not the message. But I think the critical thing is that there's so many things we have put into our food supply that are not doing us benefits in terms of our health. And there's things we've taken out of foods that are harming our health. And one of the things that has come out of our food supply is dietary fiber. And so, you know, hundreds of years ago, we ate up to 100 grams of fiber per day. And today, as Canadians, we eat about 14 grams. So we are well below the recommendations. And I think if there's a variety of foods that we can add the fiber back into, that's going to give us benefit, whatever the form may be. I have to say that the cookies tasted delicious. And the subjects who ate them for 3 months every day, they actually missed their cookies at the end of the study. So there might be some potential there.
Is there something special about pea fiber as opposed to other sources?
No, there's so many different types of dietary fibers, and they all seem to have a little bit of unique properties. They all share some common benefits, but typically there are benefits across the wide range of different dietary fibers. So we really do need a variety. If you just ate yellow pea fiber, that's not going to be the solution. So you need to have wheat bran fiber. You need to get it from a variety of sources to get the ultimate benefit in terms of health.
There's a question at the back. No, I'm calling you the back. It's actually, okay, halfway. Okay, let me count the.
Okay. So my question is prompted by what John said about cattle. We know more in Alberta about the heritage of cattle than we do about people because we don't have access to the data. But the question is really for Raylene. Is what you said is for the generic person, which doesn't exist, right? Do we have enough data to distinguish categories and classes? For example, when you talked about the gut microbiome, right? There are 3 confirmed clusters of microbiome that cross nationality, countries, and everything. So we have that data, but we don't classify Albertans into those types because we don't get access to the data to be able to target what you've learned about fiber.
Right, and that is the future of medicine, and you know, in the area, they call it nutrigenomics or nutrigenetics. So really matching up a person's genetic makeup and what their susceptibility is to disease or where they're protected against disease, and matching that up with the ideal diet. And it's not just diet in terms of lifestyle. What's going to work for them in terms of an exercise program? Does it have to be, they're going to have to be out running until they fall down every day, or could it be some weightlifting or whatnot? And so it's really matching up what is your genetic makeup, matching it with the ideal lifestyle intervention so that we can get the best benefits, so that you're not going to be a non-responder. We want to find who is out there that's going to be the responder to the particular intervention, because we don't want people wasting their time doing something, sacrificing something, you know, battling through treatments that just may not give them any benefit. So, and same thing in terms of the gut microbiota. We have our own unique profile. Each of us has a fingerprint of our bacteria that is in our intestine. And so we can give you that profile very, very quickly today. But the key for moving health and moving medicine forward is, let's now match up what your profile is to what should we do in terms of manipulating that, what's going to work best.
Maybe I'll add to that too as a clinician, because certainly right now we have to treat, and we don't know which medication will give somebody this side effect or not, but we still have to do something. So you make your best guess, that's where the art comes in. But as we learn more about profiles and things, we hopefully can also streamline so you don't have to go through some treatment that's, gives side effects and things like that.
There's a hand up here. We need to get you a microphone. Oh, yeah, you have. Okay, go ahead.
I also had a question for Dr. Reimer. There's a very popular book out there called Wheat Belly, and yet you mention wheat as one of the sources of probiotic fiber. What's the situation?
Yes, so it's a very popular book, and I get asked a lot of questions about that because wheat is a staple in our North American diet, and we've eaten it for a very long time. And so I think for me, it always comes back to what's the evidence right now to refute or to actually agree with some of these books that come out into popular press. And we know that if an individual is not allergic to wheat, or they have the condition called celiac disease, when they absolutely have to cut out wheat or the gluten from their diet, that right now there isn't really any evidence that you're going to get all these health benefits from simply cutting wheat out of your diet. Why do some people get benefit when they remove the wheat from the diet? Well, it's likely because they've stopped eating things maybe they shouldn't have had too much of in the first place. So too much white bread and lots of bagels and pasta and, just too much of wheat products. So maybe that's why they see the benefits, not necessarily that the body couldn't handle the wheat. But that's a hot topic for debate, and it gets into crop manipulation too, because there's some indication that the wheat today isn't the wheat we ate 200 years ago. So there's many facets to that argument, and right now the bottom line seems to be that it is fine in terms of health to eat it, unless you are in those very defined health conditions where it's necessary to remove it.
And the truth is, if you write a book that is full of if or maybe not and qualifications, it won't sell. And so one always has to keep in the back of your mind that a book that is a strong piece of advocacy for something is probably leaving some data aside. There's a question? Yes.
Dr. Sim, is there any relationship between these prions and Alzheimer's? Like, is there any work going on in that area?
Did you plant that question?
Yeah. I was going to ask it.
That's an excellent question, and the short answer is yes. Alzheimer's disease and a number of other neurodegenerative diseases, so even Parkinson's, Huntington's, are associated with these proteins. In those cases, it's not prion protein, but a different protein, but it too changes shape and aggregates, sticks together, and causes damage to the brain. So in that way, they are very similar. In addition, the way they spread through the brain may be very similar. And the diseases themselves are starting to be called prion-like, or prianoid, or prion-ish, or the terminology is changing. And what we are learning from research in prion disease is looking like it may have a lot of application to our understanding of these other more common diseases. So there are definitely some strong links, and there's a lot of active research trying to pursue those avenues and those links. And I think you'll hear a little bit about it later in a panel session with Dr. Cashman.
Yes.
My question's for Dr. Sim. In one of your slides, you showed infected brain tissue, and then you showed, right beside it, with treatment, some recovery. Right. So what mechanisms are there for recovery, or what potential treatments?
Right, so that's an excellent, and I should clarify, so in that specific picture, there were two separate brain slices, so it wasn't the same slice that then recovered after treatment. So that's an important point, because what we do know about treatments in these diseases in particular, the earlier you start treatment, the better, and once you get past some point of no return, the likelihood of seeing that benefit can be lost. And there are several approaches to trying to treat these things. One is to target that misfolded shape itself. So you can either try to prevent the normal protein from becoming the wrong shape. You can try to take the one that is the wrong shape and get rid of it somehow, but it's hard to get rid of because it's very resistant to most methods. But try to prevent it from getting worse or getting bigger. So a lot of research has looked at trying to use what we call conversion inhibitors and blocking this. And while that sometimes works in the test tube, it doesn't work as well in the animal, and then the few that have made it into human trials, they really don't pan out. But again, the caveat there is by the time the treatment is started, often a lot of damage has been done. But another approach is to look at, well, what is the cascade of events in the cell that is triggered to cause the holes in the brain in the first place. So can we possibly target that shape shifting keep that under control, but at the same time use a combination therapy and hit the point where those cells are starting to die and try to help them deal with the stress of all of this extra protein. And I think that's where a lot of people are looking now, is to try to combine those two. But it's difficult because just picking one particular treatment approach, it doesn't work. You say, oh, well, that's no good, and you try another one, and that doesn't work. I think we really need more than one target at the same time.
We do have so we have time for one more question, because the stunning thing is we're actually on time so far. And we're also, unbeknownst to all of you, monitoring the caffeine levels in your brain, and they're dropping. So we'll have one more question, and then we're going to have a 20-minute break for coffee. And if you have questions for any of the three speakers so far, you might be able to engage them at that time. So, yes.
Just a quick question for Dr. Sim. Has the Prion Center in Alberta, I heard that there was a possibility that funding has gone away. Are you still receiving funding for the Prion Center?
So there's a Prion Center in Edmonton. So we're called the Center for Prions and Protein Folding Diseases. Nice long acronym. There's also a Prion group in Calgary. So they have a center as well where they can, it's smaller, but they can also do Prion work. We have been basically supporting the center through the primary investigators. So each of us then seeks funding through our partners, through AI Bio and stuff, and through those funds are supporting the center itself. Certainly that's always a question because some funding comes through support of the university, some from the government, and of course a whole other topic for discussion is funding of universities right now. And universities, and U of A in particular, of course, has had a lot of those cutbacks. So certainly we, in addition to all of the other groups at the university, have fell on hard times with respect to funding, but we are still extremely active in writing grants and putting out the call for funding. If you're offering, we would love some. That would be great. But yeah, I think for all researchers in this climate, it is of concern. And I think these events are important to try to show you the purpose and the point of it, not just from the final applications, but right at the ground level, because without that entire continuum, you can't get to that end point.
I live in southern Alberta, and obviously one of the biggest event of the last year was the flood last June. And, it says something interesting about how we Albertans and visitors to Alberta are changing the landscape. And it's true that the flood seemed to have been a result of three unusual situations happening at the same time. A storm cloud that sat in one place and dumped more than a foot of rain over a brief period of time, a snowpack that was still extensive but was melted by that warm rain, and a high water table that didn't allow that water to penetrate into the ground. But part of the issue with the flood, or at least an issue that was highlighted by the flood, was the fact that the Bow River Valley has been changed permanently by human recreation, agriculture, forestry, oil and gas. And I think it brought to our attention the fact that whether we think about it or realize it, we are changing the entire landscape of this province, and we need to be able to track those changes if we want to retain the biodiversity that, in fact, all of us love to have around us. Now, one of the principal areas where this is crucial, not just the Bow Valley, is of course in the northern part of the province in the oil sands. And that is at least partly the focus of Scott Nielsen's research. He's Alberta Biodiversity Conservation Chair and Associate Prof in the Faculty of Agriculture, Life and Environmental Sciences at the University of Alberta. Scott.
Thank you, Jay. Let's see if it'll advance. Whoops. I'm going to talk a few minutes about some of the work we're doing in my lab relative to the challenges of biodiversity conservation in Alberta. And in particular, as Jay was mentioning, rapid changes in land use, climate change, other things that threaten our natural heritage, our biota. One of the primary challenges that you hear in the media quite a bit, both locally and of course international attention, is the oil sands. The map on the left is the oil sands, the bitumen deposits. The darker colors in the red and maroon are deeper deposits. Some of these deposits are close enough to the surface to allow open surface pit mining, and that receives quite a bit of attention. But I argue from a terrestrial biodiversity standpoint, our greater threat in the future is the in situ. So that region represents 142,000 square kilometers of bitumen deposit. Only 3% of that is close enough to the surface to be mined through open pit mining. 97% of that has a potential of in situ mining. So with in situ mining, they're drilling wells instead, and it changes the landscape in a different way. It may relate to only about a 10% loss in total habitat, but there's a tremendous amount of fragmentation that's associated with that type of mining. This is an example image of one of these leases. You'll note here, the first thing that is done is seismic lines are first cut through the forest on a grid. Oftentimes, in some places, these are on 100-meter or even less spatial pattern. And these are used to essentially map out the reserve, the bitumen reserve, to know the depths and for drilling. Next follows the well pads and infrastructure and pipelines and other type of activities that are used to extract and move bitumen. The issue with this is this changes the landscape. This changes the boreal forest tremendously. You have winners and losers, and in fact, one of the major issues is these seismic lines and other linear features increase movement of organisms. In some cases, that is to the detriment of some species like woodland caribou. So here, these seismic lines increase primary prey for wolves, so moose, deer, they increase in abundance. The primary predator of moose and deer being the gray wolves also increase. And it turns out that the seismic lines and other linear features also allow greater permeability and movement of wolves. And that increases the chance that wolves bump into caribou. Caribou numbers are quite low, and with increased predation rates from wolves, caribou populations have been declining dramatically. As a response to that, Environment Canada, they've been classified as a threatened species under the Species at Risk Act. With that, there's been a recovery strategy, and the recovery strategy has identified to leave 65% of caribou range in Alberta as undisturbed. And what they define as undisturbed is anything that's further than 500 meters from a human footprint or disturbance. So you'd basically take this entire landscape you're seeing in this image, and remember those seismic lines are about 100 meters apart, a 500-meter buffer on all of those. So essentially you're seeing in the boreal forest that there is very few areas of undisturbed habitat for caribou. That has created a great challenge because if they establish a threshold of 65%, something has to be done to recover habitat. One of those is restoration or reclamation. So there has to be some way of recovering these seismic lines. I have a student, Cassidy Van Rensen, who's working on identifying patterns of recovery, of restoration of seismic lines, and approaches for reclamating those. So the image on the right here is an example of silvicultural techniques of basically increasing microsites, mounding the soil, reducing compaction. This is typically used in the forest industry, but we're trying to find ways to increase establishment and growth of trees on many of these seismic lines that are not recovering. Her study area represents a small area of the oil sands in the in situ region and is southwest of Fort McMurray in the Stony Mountain area. In that area, there's, in the broader area of the Stony Mountain, there's 12,000 kilometers of seismic lines. In the sub-study area that she's working on, approximately 1,300 kilometers of seismic lines are unrecovered. So they're in a form of arrested succession. They're not recruiting trees and growth and facilitating the movement and the changes in the composition of the boreal forest. The restoration techniques are expensive, however, about $4,000 per kilometer to restore these seismic lines. So in her small study area, that would be almost $5 million to restore habitats of this, for woodland caribou. Well, that's quite a challenge, and probably not something we're going to see those type of investments in conservation. So we have to find a way to identify some areas that are naturally recovering. It doesn't make any sense whatsoever to put $4,000 per kilometer on the few places that will naturally recover itself. So we have to find a way to prioritize the restoration actions given the limited resources we have for restoration and conservation. So the approaches that Cassidy has been using is targeting, an example would be, let's target 50% recovery of seismic lines in this study area. And let's try to consider the fact that some places will naturally recover. Let's consider the fact that let's emphasize caribou zones. So the map in the upper right, labeled A, those polygons are different areas of caribou zones and different levels of priority for conservation, with the yellow and orange areas being the most important. And we should also emphasize the fact that we might as well avoid areas of high bitumen value. So in B, the areas in red there have greater bitumen depth. These are areas that we probably shouldn't put any restoration efforts. They're going to be highly active today and into the future. So we're essentially trying to optimize the location of where we put our research dollars and our conservation dollars for caribou restoration. So in this example, using an optimization algorithm, she was able to find a solution for restoring 50% of the seismic lines for $1.8 million, rather than randomly a cost would be $2.44 million. So more than $500,000 savings in one small area. The second topic I'd like to talk about is, well, we have more than just caribou. We have, our biota is rich in Alberta. If you take a look at some of the other rare species we have, in this case, rare plants, bryophytes, lichens, and butterflies. This map on the right is a hotspot map. Areas in red represent areas of higher biodiversity for rare species. So you'll notice that the mountains in particular, the foothills, some of the grasslands area, many areas of the moraines near Edmonton, all of these areas have fairly high biodiversity. Parts of the boreal forest, less so, but some places such as McClellan Lake up in the oil sands are quite important. Now the challenge is, how do we find the places to best represent and conserve these species? We already have a protected areas network, and that protected areas network represents quite a bit of those species in the mountain parks, wood buffalo, but the protected areas weren't designed necessarily for biodiversity. They were more designed for recreation and tourism. So what we've done here is I've taken that biodiversity hotspot map, and I said, let's protect 10% of all the species. And if you can't protect 10% of those species, where are the most important places in Alberta to meet that goal? So there are, of those 308 species that I've been working with, there are places that they are not represented in these different type of protected areas. The areas in orange and red, the yellows, these are areas that we would consider highly irreplaceable. Those are the places that would best be able to achieve our conservation targets of protecting 10% of Alberta's rare species. So you'll notice areas like the Beaver Hills here, South Saskatchewan, those type of places are quite important, and they're not represented in our current protected areas network. The second point of this is, if there are major developments such as oil sands, we could use these same concepts to identify biodiversity offsets or conservation offsets. So given that there's disturbance in some areas, where could we put some conservation dollars to protect some of Alberta's biodiversity? Third topic I'd like to talk about in the last one is the fact that the world is changing. There's this exogenous factor of climate change. It's not just the land use, and both of those interact and have effects on the persistence and the viability of our biodiversity. So this Quinn Barber, a student of mine, is looking at climate velocity. And this is how fast climate change is moving relative to your ability to keep up with climate change. So in red areas, this represents areas of high climate velocities. What this represents is if you're in the top of the mountain or you're a flat expanse, if the world warms by 3 degrees and you have to maintain your current climate niche or habitat, how fast do you have to move in kilometers per year to keep up with climate change? Well, it's anywhere between 0.1 kilometers to 10 kilometers per year. So areas in red, you have to move 10 kilometers per year just to maintain your original or existing climate niche. Now, it turns out that every place is different. If you're in the mountains, or in the foothills, I should say, you don't have to go very far. You can go upslope. So you either have to migrate to more northern latitudes or to higher altitudes. And so part of this is buffered in terrain, areas of terrain, except for in the top of mountains. With those places they can't necessarily, they're migrating to, they have to make major jumps to different mountaintops. So you'll see the hill systems in the boreal, the grassland areas, and some of the mountaintops in particular are going to be quite sensitive to climate change. Now, if you also consider habitat fragmentation, if the species have to migrate, they have to move through a landscape. So we'd have to understand that land use change as well as climate change and the ability of these species to move to keep up with climate change. So in red, these are areas of high habitat loss and fragmentation, the parkland natural region, some of the grassland natural region of Alberta. These are the most fragmented places. And some of those places are also the places that have the highest climate velocities, creating quite a challenge for species conservation. On the left side is a graph representing a couple hundred rare plants in Alberta, and these are graphed relative to those 2 dimensions, climate velocity and habitat fragmentation. So some species occur in areas of low climate velocity, low habitat fragmentation. We're not necessarily so concerned about them relative to, let's say, climate change and land use changes, at least existing land use changes. But other species, we would be quite concerned about if they're occurring in areas of high habitat fragmentation and high climate velocities. So one of these species I'll pick on is longleaf bluets, Houstonia longifolia. A rare species in Alberta, only occurs in about 20 locations. It occurs in an area of high habitat fragmentation and high climate velocities, making it quite vulnerable to future climate change. One of the things we're doing is we're taking that particular species, as well as another species in similar habitats called northern blazing star here, and we're using proactive conservation tools to address these kind of challenges for these species by using assisted migration to move these species north. So these point locations are the known populations of northern blazing star and longleaf bluets in Alberta. They're occurring in those areas of high habitat fragmentation and climate velocities, and we're exploring the approaches for assisted migration. With that, I'd just like to acknowledge some personnel in my lab and collaborators. This work is being done under the Alberta Biodiversity Conservation Chairs Program with Alberta Innovates Biosolutions, COSIA, and a number of other partners, looking at the challenges of managing the future of Alberta's biodiversity. Thanks.
Thanks, Scott. I began this event by talking about what other people in Canada think about Alberta. So I can turn that around and say, well, what do Albertans think of first when they hear Toronto? I mean, I don't even have to say it. Will Harney lives in Toronto, but I want to make a really important point. He works in Vaughan. The mayor of Vaughan is Maurizio Bevilacqua, so he's okay when it comes to mayors. Will works with Magna, the car parts manufacturer. You know, my son came out here this summer to work, and I had to buy him a beater. So I bought a 1997 Chevy Blazer. which is in actually remarkably good shape. But it is immediately obvious when you sit in this car that cars have advanced dramatically, especially when you go to fill the tank after a day and a half. And when we think of the automobile and sustainability, we do think of gas consumption. That seems to be one major factor. It's now listed as one of the attractive options in most new cars. But Magna is interested in sustainability with respect to car manufacture in a completely different way, and that is using biomass. So, Will is Executive Director of Research and Development at Magna Exteriors and Interiors. Please welcome him.
Do I say thank you or thank you? Well, again, thanks to Stan and AI Bio for asking me to come out and chat with you today. It's wonderful to be in Alberta, and it's even more wonderful to know that the province has a history, long, not a lot of data, but a history of taking colorful city mayors and turning them into fantastic premiers. So maybe King Rob's got a nice ring to it. All right, as Jay mentioned, I'm with Magna International, and I'm here to talk about a project we're involved with AI Bio, and it really focuses maybe a little bit differently than the other speakers on much more entrepreneurial and much more consumer-focused activities. And so we'll use an automotive icon, a sign, to get things rolling. But I think the key piece for us is, it's very true. What Jay said, we have an incredibly different environment today for the nature of the product we're driving and buying. And it's being influenced massively by these mega trends, which affect actually everything that we're even talking about today. But I'll key in on a couple of them, and this really drives our role as a supplier. We're a supplier to the automotive companies. We do it a fair bit. And more we're being involved in designing, developing, engineering systems, products, features, and in fact architecting materials to provide those systems. So we look at trends like globalization, demographic change, of course scarcity or more distribution challenges with fossil fuels, and how does that affect what we do as producers of components and subsystems. Also very much interested in looking at demographic change. It changes many things. Obviously as the population in developed nations or regions age, you have factors. It also shapes how companies position themselves to exploit those markets. Jay mentioned his son. I can attest to the fact that I have millennials that don't buy cars. In fact, they won't leave my basement. So Magna, in our quest to be the greatest and most innovative automotive parts manufacturer on the planet, has three primary strategies and priorities. One is world-class manufacturing. Second is people development to fuel this change. And the third is innovation. And when we look at innovation, of course, not uniquely, we look at innovation across a number of different attributes. And a key point, as Jay mentioned, is sustainability. We look at sustainability from the standpoint of economic sustainability as well as environmental. And really, at the end of the day, it drives down to reducing your input costs, which more and more are energy costs, and therefore it behooves all of us to find ways to identify lower cost energy sources, which oftentimes are sustainable. Schwack, beautiful. Kind of a sort of a tone-setting piece is globalization, and it can mean a number of things. But today I want to talk about it, I guess, in two ways. It also means for me today being on a 6 o'clock call to Germany and then having to be around for a 10 o'clock call to Japan, but that's my problem, not yours necessarily. It also means that our customers, and these are the large, highly integrated global car companies, are necessarily globalizing their product range to reduce engineering costs and to be more effective at providing product for various regions. And this is not, the global platform discussion is not the only discussion. There are other very, very important regional discussions, like, for instance, pickup trucks, which are an important economic driver in North America for obvious reasons. But in the case of the Golf platform, Volkswagen Audi Group, basically creates what are called top hats that go on top of the platform that's global to provide products into Spain, Czech Republic, North America under badges like Audi, Volkswagen, Skoda, et cetera. And so when we work with Volkswagen to provide technology for them, they say, great, I love it, but can you provide it where we build our cars? So it's a key piece, and it's not a big company diatribe to say that you have to be everywhere, but you need to be everywhere. So when you look at technology development, especially around materials development, you have to take that into consideration. How do you provide a uniform product delivery across the entire globe for a new and innovative product? So if you don't have a regional play, you've got to consider this. So we look at that as one of the many drivers. We also look at some of the facts, and it was touched on earlier in the intro. Obviously fuel economy regulations are increasing faster in China, mind you, than they are in North America, but nonetheless they're increasing. And that's huge. The 54.5 mile per gallon is the iconic US fuel regulations, which are coming into play in 2017 and mature by 2025. Europeans consider more in terms of CO2 produced per kilometer driven by a vehicle, so CO2 regulations. Recyclability, very, very important. Today, the standards are approaching mid-90s for recyclability for the end-of-life product. And then key for me also is the $9.95 price piece. Because in Canada this spring, you'll be able to buy a Nissan Micra with a MSRP of $9,995. And that's a car with 5 doors, tons of airbags, steering wheel, all that good stuff. It's incredible. And not only is it a safe car, but it's not a car that's being dropped into a market to dump and try and win business. It's a major automaker's play to try and expand their range into smaller cars. It's legit, big time. So all that said, the challenges around weight reduction, which means typically they're in lighter materials or fewer materials, but you can't make smaller because people won't buy small. Consumers ultimately decide what they want. I want a pickup truck, I want a sedan, I want an SUV. It's got to do things for me. I'm not going to, downsize my needs to suit some regulatory requirements. So the challenge is to make these vehicles provide all the functions and features, but way less so they use less fuel. And that's why when DuPont asked engineers and designers a few years ago where they thought the shifts were in materials going forward, off the charts was advanced composites. So the idea of combining fiber with plastic, basically, and creating composite technologies like Boeing's using for the 787, whether it be carbon fiber or glass fiber. This idea of making plastic punch above its weight through reinforcement is a key strategy for weight reduction. So if we look at fiber-reinforced materials, in this case, biofiber-reinforced materials, and typically that means an olefin like a polypropylene and a fiber, we're seeing a growth, as predicted by the study by DuPont, in these types of materials. And if you look at even today, a snapshot of 60 million cars produced and sold globally with about a 14 kilogram payload of these types of materials per car, it's a significant amount of material already in terms of just a current market size for this kind of material. Oftentimes, they're not global, but they're regionally applied technologies. Our goal now is to try and take that farther. So as an example of where this trend in fiber-reinforced products is going, again, just blowing our horn for a second, we launched this year, or actually last year, 2013, a full olefinic, meaning polypropylene-based, fiber-reinforced liftgate assembly for Nissan, for the Rogue vehicle. Now, this stuff, plastic liftgates is not, cocktail party talk, for sure. But you're a captive audience, so I can I can take advantage of this. So it's a big deal for us because it's a first in North America, first in NAFTA. Nissan builds 170,000 Rogues. That's 730 of these things going out the door every day from our operation. And it's basically representative of this new technology, fiber-reinforced plastics, in this case glass-reinforced polypropylene, that's setting the stage for new types of paradigms for these systems. Provided Nissan with a weight saving and a cost saving, so that's what they want to hear. So all that said, we're a big user of plastics, as you can imagine. This is a quick snapshot for 2014. Approximately 225 million kilograms of plastic, resin, olefin, what have you, nylon, various other plastics. So we pay a lot of attention to this because it's a big deal for us. And if we start reinforcing these plastics, we have interesting new materials. I think going back to the other trend, which is the whole energy or the fossil fuel scarcity discussion, There are shifts, as certainly you well understand, regarding what types of fossil fuels are being harvested or extracted, whether it be shale gas or tight oil or bitumen-based oils and so on. And what that's changing for us is a change in how the co-products around refining these inputs is changing. And that directly impacts the economy around these different materials. So it's a bit of a moving picture all the time. So we buy a lot of plastic, and we buy a lot of plastic from some very, very large companies. And you'd think for us, automotive is a big deal, and it is. But for the guys and girls who own and drive these companies, automotive is not a big deal. It's important, but it's not a big deal. On an aggregate basis, all these large petrochemical companies typically sell less than 1% of their global turnover into automotive. So, we think we're a big deal, but they don't. I mean, they like us and everything, but we're just not a big deal from a research perspective. They won't invest necessarily the kind of energy into innovation that we would like to see, because for them it's not that important. Touching on demographics, I mean, last week or a week and a half ago, Bayer announced that they were selling their plastics division so they could focus on pharma. Because they're seeing demographically it's more important to buy and do R&D in pharma, not so much in plastics, and certainly not so much for cars. So we've got a problem. Big demands for lower cost, lower weight, more sustainable, innovative materials, and we're kind of a little bit of on our own in a sense. Which is why we kind of found ourselves here, literally and actually. If you look at reinforcing plastics with fibers that aren't necessarily glass or carbon, inorganics, but look at organic fibers, it opens up a lot of opportunities for some pretty interesting material solutions. And it addresses some of the key drivers we're seeing from a megatrend perspective. Fossil fuel constraints, material shortages, and then, of course, demographic change as we trying to deal with different consumer behaviors. So today, we're talking about, very briefly, the work we're doing under AI Bio on wood fiber reinforced polypropylene. Basically, that's the deal. So we see this as a way to create not only a new material solution that's going to be lighter and cheaper potentially, but also one where we can get the kind of necessary research focus to actually create specific material platforms to generate the product functions that our customers need. And this is a very, very brief snapshot of what we're talking about. But essentially, we're trying to work to go right from forestry products into car parts. And what we know a lot about is on the right-hand side of the screen, combining polymers with fiber, designing systems, developing process technology, scaling it globally. But as we go to the left of the screen, we know slim to nil and slim's not home. So what we found in this partnership is a deep resource of research knowledge and engineering knowledge around fiber treatment, because at the end of the day, it's the fiber interaction with the polymer that makes it or doesn't make it. So that's our conceptual process path, and that's the work we've been doing over the last number of years with AITF and our partners. So the good news is if you're successful, and we think we are, and we will be more so going forward, what you end up with is a material that competes with essentially the blue bar on the left, which is a fiberglass reinforced polypropylene, standard Army issue material used for almost many car parts. You can get similar performance in terms of physical performance with a wood fiber reinforced component, and it has a lower density than the glass fiber reinforced system that it replaced, which means fundamentally for the same amount of work, you have a lower mass part, which is what drives to lighter vehicles and happy, low CO2 consuming customers. There's also some upside on cost because the fiber itself is lower cost than glass fiber, which translates into economic gain to allow us to build these 9995 Nissan Micras. So just again, throwing it back to my fellow speakers, just to thank the people who are involved in this process. We have a lot of work to do, a lot of it in the front end, which again is an area we don't have particular expertise. We found that expertise through our partnership. We have our friends at the Center for Research in Bioeconomy supporting this project. AI Bio, as we discussed, supporting the project. We've been working deeply with AITF on the fiber piece and some of the early compounding studies and doing the screening of different pulp technologies. And we have support through the NRC, who is basically a roommate at our Composites Technology Center in Toronto. So again, just going back to how we can use this, I think the original topic was secret ingredient to automotive technology advancement. We can see that by harnessing biomass, sustainable, globally relevant, repeatable wood fiber technology, we can address some of these major concerns, megatrends, and be positioned properly to provide solutions going forward to satisfy consumer needs. All right, that's it for me. Thanks very much.
Can you just stay? Yeah, right over here. We're going to get the questions for you. And Scott, could you come back up? So we're just going to do the 10 minutes Q&A from the audience to either Scott or Will, and then we will move on to our session. And there's an arm up at the back near the aisle. And then we'll move on to the scientist entrepreneur section of today's presentation. And while they're getting out, could I ask you a question, Scott? First, I'm not sure you used the word triage, but it certainly sounded like that's what you were talking about. And is that the kind of era that we're in right now, where we have to accept that some things can't be saved?
Yeah, I'd agree. So one of the issue comes down to we have limited resources and ability to conserve or maintain some of our species, perhaps woodland caribou being one of them. And at some point, do we invest those conservation dollars to try to do something where we probably have a low chance of success and triage that, and instead concentrate some of the conservation dollars to things we can save? So I think that it'd be quite controversial, but that is possibly some of the next phases of discussion in conservation.
Wow. Yes, question.
My question from the back is for Scott, and it's about the measurement of climate velocity.
Sorry.
So what I understood is you measured climate velocity independent of the movement of the rare species, but do you have an idea about how to do exactly the inverse by building baselines from movement of species, because they don't always move because climate change, right?
Yeah.
Geography, geology, and other things. Do you have baselines that help support the idea that your climate velocity measures are accurate enough to optimize to the experiments you do?
Yeah, that's a good question. So the climate velocities that are described in those slides assume that a species needs to maintain a climate equilibrium, but that is an assumption. An alternative is in situ adaptation. So you'd adapt in place, and that climate itself would not be necessarily a limiting factor. So this is perhaps an exposure type variable, but it doesn't necessarily absolutely suggest that those particular species are most at risk. So the next phase really is, as you describe, is looking at dispersal capabilities or dispersal kernels, the natural dispersal abilities of species, and further sorting down those species to those that have very low dispersal capabilities and perhaps have a narrow climate niche that aren't so tied to, let's say, an edaphic or soil condition. So certainly that's one element of possible vulnerability or exposure to species.
Yes.
Hello. The question is for William about wood fiber reinforced components. One of the fibers that's interesting is the cellulose nanocrystals, and I would like to know where you see the opportunities and the challenges for those.
Okay, so wood fiber nanocellulose is not in the scope of this project initially, but where we see the application would be in modifying the core polymers to provide different coefficients of thermal expansion, toughness, and other attributes which would improve the if you will, the matrix, not the reinforcement. So this technology is 100% focusing on fairly large volume fractions of wood fiber in the final composite, meaning on the order of 50%. And NCC, I think, has an enormous opportunity going forward relative to modifying resin systems to adapt its behavior. Our challenges that we see right now are mostly in the area of dispersion technology, understanding how to integrate or disperse different dispersion, I think. Dispersion of the NCCs within the matrix itself.
And toward the back there. Yep.
Scott, a question for you. Regarding your work in restoration activities and monitoring restoration activities of the forested areas and surveying the caribou migration patterns, have you ever considered using unmanned aerial systems for observation purposes and tracking purposes?
Actually, that's a good question. There's two elements of that. One I didn't describe, but we are using LiDAR, so existing remote sensing, high-resolution remote sensing information that maps the canopy and forest stand, so we can look at areas without actually actively going in the field and measuring the vegetation height and trees and so on, which is fairly time-consuming over the broad spans of the areas we're working on. So we're using LiDAR to figure out areas that are recovering or not, understand what types of features in the landscape that promote natural recovery versus promote that arrested succession state, and then given that, target the restoration actions. And we've just been starting to use unmanned aerial vehicles, or UAVs, and drone-type UAVs where we can send on missions to send down seismic lines or map well pads and for reclamation purposes. So I think that has a lot of opportunities. It's much more of a small scale type thing, but they can be used every year or every 5 years, and they can actually monitor vegetation recovery on some of these places where it's important to restore.
And this will be the last question for this round, please.
Scott, in measuring biodiversity, are you also looking at the metadata, the microorganisms below the soil? Anybody capturing that?
Not myself. So I don't know the answer to that question. I think in general, not much is done in that area, so it certainly is an area of future possibility. Currently, the public puts a lot of value into some of the more charismatic species, flowering plants and large mammals and things like that. So there's pressure to sustain populations and the viability of species like caribou, but not much work that I know is being done in the boreal forest on that topic. Yeah.
Great. Thank you both. And so this year we've added a really interesting feature on scientist/entrepreneur. I think many people outside of this room would think there's not much of an overlap between the typical entrepreneur and the typical scientist, but there can be, but it does require a special skill set, and we have two outstanding examples, both of whom will tell you a little bit about their experiences. So first I'd like to, well, I'm going to ask them both to come up, actually, yeah. And one will sit while one talks, and then we'll flip them around. So first, Dr. Jackie Shan, she's founder and chief scientific officer of Affinity Life Sciences Incorporated. She might be the only scientist who actually can say that Don Cherry at one point was one of her colleagues. Jackie. Do you want the slide changer? It's over there.
Thank you very much, first of all, and thank you for the honor to invite me to attend this really very, very interesting session. So I'm just talking a little bit about myself and then the business I have created and some of the challenges associated with being a scientist turned into the entrepreneur. So the first company I co-founded was CV Technologies, and later on renamed as Afexa Life Sciences, and it was a spin-off from University of Alberta Department of Physiology. So it's almost there to 20 years, and really what the business was about, it is considering kind of a pioneering. We're trying to develop and commercializing evidence-based natural health product and for disease prevention and health maintenance. So what we do is we're isolating the natural compounds and from a variety of natural sources, herbs and food sources, and trying to develop that into a therapeutic product or a consumer health product. So we've been there for almost 20 years. We all know at the beginning, I just talked a little bit challenges for some of you and the scientists who want to actually become an entrepreneur, get involved in the business. So really it's to summarize just my experience in the last 20-something years. So it's highly risky, it's highly rewarding as well, and then later on you probably know the highly I'd say, a branded product called FX. So that was a highly and rewarding. However, during this process, it's highly risky, the time and effort and cost. And in our cases, because we're doing the consumer branding ourselves, so there's also associated with reputation risk. So not everything said about you is good. So, and then challenge is also being a scientist and a science-based business. I think everyone has to have this, and the challenge of integration of a science and a different discipline. So we have pharmacology, we have a chemist, and we have medical research and clinical. So you're trying to integrate them and you eventually become a business, and then let alone the business team, the sales and the marketing and the branding team. So it's talking like a foreign language. So that's one of the things from operational is a tremendous challenge for us, but it's evolving, eventually it gel together. So, however, it's highly rewarding. And so we spent in the first 10 years creating the technology, trying to be competitive in the marketplace. And the technology to ensure we have patented and proprietary technology, but also come out the competitive product. So CodaFX is one of the product, and we're very proud that we'll be able to start from scratch, eventually bring to the marketplace and brand it ourselves and become #1 cold and flu and product in the market in Canada. So that's something we're very, very proud. We're all very in Canada, we're not very known about as a branding to build a brand, but our product eventually beat up a product like Tylenol, and from the retail sales point of view. So it's that is highly rewarding. And in addition to CodaFX, of course, that's people know, we also develop a program in cardiovascular. So we're in the clinical studies and searching for diabetes and hypertension and lowering the cholesterol. And we also have a product in the cancer research stage, and also have a product for central nervous system. So I'm just talking a little bit about the CodaFX, and a lot of people know about Don Cherry and the brand. And however, being a scientist, that we stay firmly and grounded with on the science. As you see, I'm just summarizing the clinical studies and we've done before and after we're actually launching the product. So it's pretty sizable, and considering a natural health product. So we invested about $20 million and did about over more than a dozen clinical studies, and it's continuing. The study is continuing, and they're trying to branch it into a different specific population, senior and some in the children. So just a little bit of summarizing the fate of CodaFX. So in today's world, we all know everyone is buying everyone is merging with everyone. So CodaFX, and actually, I don't know how to put that, whether I'm happy or I'm sad. So eventually got bought by the Valeant Pharmaceutical, international pharmaceutical, its largest pharmaceutical company in Canada. So that was 2 years ago. And being a scientist and myself, my passion really, even though CodaFX being acquired, and I really like to continue this journey about, being a scientist. We have a lot of innovative ideas and the technology, not only the CodaFX. So I like to continue to my journey. That's the reason I created another company 2 years ago. So just very happy. And recently, the company in the 2 years, we start launching the product already in the drugstores, in the Rexall, and also in the Values Drug Mart today. So we continue doing the research. And recently, I'm also negotiating getting all the technology, all the pipeline product back from Valeant Pharmaceuticals. So in a way, it's a kind of a happy ending. Right now, they actually work with me, trying to help and to see how can we, pushing more new products in the marketplace. So just a little bit of promotion, and then I was very depressed that once the CodaFX was acquired. In 2 years, actually first years, I said, well, one of the coping mechanisms for me, instead of being a desperate housewife, I can write something. So I actually put something in a book, and recently just launched it last month, and the book actually is selling right now in the Indigo and the Chapters and the Amazon.ca. So it's a little bit coping. Sorry about taking a little bit more time. Thank you.
I know Neil Cashman pretty well. He was instrumental in the formation of PreonNet, one of the National Centers of Excellence. For reasons that still escape me, he invited me to be on the board. I sat there for the board meetings and marveled at all the intelligent people around the table. Neil was preeminent among them. So he's a prion researcher, he's a neurodegenerative disease researcher, and please welcome him. That was great.
Well, I was going to, and are people hearing me? Yeah, I guess now. I was going to say what an honor it is, how proud I am to actually speak at this function. It's really, Alberta is doing incredibly well with innovation, and I'm lucky enough to serve on the AI Biosolutions Board of Directors, and I've just been impressed and impressed and impressed as time goes on. So I was going to start with that, but I think instead I'll start with saying how hard it is to follow Jackie. I really feel kind of inadequate in several ways, but I'll try. In addition to starting companies and being a scientist, I'm also a physician, a neurologist, and my lifetime clinical obsession has been amyotrophic lateral sclerosis. Does anyone in the audience know what that is? Can you raise your hands? Okay, so about half or so. It's so everyone knows, of course, what Alzheimer's is. Everyone knows, of course, what Parkinson's is. But fewer people know what ALS is. It's another neurodegenerative disease, and I'll show you some information about that and how we might be able to approach it. So another thing I like to present with my talks, you know, that we're always required as academic researchers to put up our disclosures, our industrial disclosures. Well, that sounds so furtive, like you're trying to hide it and somebody's forcing you to bring it out. So I changed the word to engagement. It also gives me the chance to say that I actually founded 2 biotech companies in Canada, both of which are publicly listed. I have provided, I've licensed a technology to Biogen, also to CanGene, which has now been bought up by Emergent Biosolutions in the United States. And I serve on the scientific advisory board of Prothena Biosciences in the Bay Area. So I want to really summarize this in 2 slides. This is my intellectual passion, but if you understand these 2 slides, then you understand what I'm doing and why I'm doing it. I think we all learned in high school that proteins are a chain of amino acids, but maybe we learn in university or not that this chain of amino acids actually is folded into a precise 3-dimensional shape in order to achieve the function that it was designed for. So this doesn't have any function as a protein. It's just a floppy string. This has a function when it's been folded properly. It acquires its function. Maybe it stops there in our training, but over the past 10 years or so, it's becoming increasingly clear that proteins can become misfolded in a kind of propagated pathway, almost like, as Val Sim was showing, a chain of dominoes. One bad protein convinces the protein next to it to convert into more of the bad kind, et cetera, et cetera, et cetera, till all of the protein in a brain or a tissue is converted into this abnormal toxic form. Of course, that idea came from the prion world, from Stan Prusner's discovery, sanctioned by the Nobel Prize, that the prion protein is an infectious agent because it acts in this domino fashion, templated at the beginning or spontaneously misfolding or genetically determined. Once the seed of misfolded protein is formed, it gets propagated. But surprisingly, or maybe not maybe not so surprisingly, and Val mentioned this prion idea is starting to find application in common neurodegenerative diseases. There's a prion-like component to Alzheimer's disease. There's a prion-like component to Parkinson's disease. There's a prion-like component to ALS. There's a different protein in the starring role of all of those diseases, but the concept, the process is the same. We're also exploring the idea that protein misfolding may not be global or permanent, that there are regions of proteins that can partially unfold in a kind of dynamic process, unfolding, folding, unfolding, folding. And this may achieve significance in the treatment of cancer. And I won't go into a lot of detail about that, but I'll show you the second disruptive idea that my work and my companies are trying to advance. And that's the idea that antibodies are perfect golden bullets or silver bullets for these sorts of diseases. We're kind of all raised in the scientific world to believe that if we only turn it over to Pfizer, they'll find a small molecule that fits in a precisely defined pit or cleft or channel or something like that, a rigid target on the surface of a protein that can be bound by a small molecule, also itself essentially rigid. But for protein misfolding, that's the wrong pathway. Protein misfolding is characterized by large floppy regions of proteins, not a little defined pit or pocket, but a floppy region in which small molecules are essentially useless. But an antibody can be designed or selected to specifically bind to a misfolded portion of a protein. It's like, this is what antibodies eat for breakfast. Small molecules can't do anything about these floppy regions, but antibodies can. And there's an advantage also with antibodies that they may neutralize the toxicity of a misfolded protein. We know that they're essentially all toxic. They kill cells. They may block the propagation of misfolding. They may accelerate degradation of the misfolded protein. But there's a safety aspect to this idea as well, that if you're leaving the normal protein alone, sparing the normal protein, you don't interfere with the function of that protein. You don't have widespread whole body autoimmunity, all of the places where this protein is found. So we have, we now, based on these two ideas, have, okay. We have a vaccine under development for chronic wasting disease. As Val was mentioning, it's spreading like crazy in the wild. We don't have a treatment or prevention, but there is a possibility this vaccine against partially misfolded prion protein could provide that protection. We are exploiting at Amorphix, the company I started, Amorphix Life Sciences. The initials, of course, are ALS. We have a raft of specific immunotherapeutics for cancers which spare the normal cells. Again, this same idea of leaving the normal cells alone and only targeting the sick cells or the bad guys. We have a therapeutic for Alzheimer's disease in development by CanGene, which recognizes a specific immunologic target at the surface of A-beta oligomers. Do I have a few more hours? Okay, I guess so. And finally, we have we've recognized that superoxide dismutase 1, SOD1, there will be an exit test, is a potential target in the treatment of ALS. If we can stop the cell-to-cell spread of SOD1 propagated misfolding, then we might be able to arrest ALS. This technology was developed through Amorphix, but is now under development by Biogen. So quickly, in such a simple way, even I can understand it. Here's protein misfolding, a sick protein inside a cell, which is converting its neighbors, going to a neighboring cell, and converting those normal proteins and producing cell death. So if we have an antibody or a vaccine, the same process occurs, but it's blocked in the space between cells and the nerve cell is protected. Just one more thought. Out of the Big Bang of the prion protein and prion diseases, there are now tie-ins for not only Alzheimer's, ALS, and Parkinson's, but type 2 diabetes, Huntington disease, amyloidosis, and even perhaps schizophrenia. It's a brand new world. And I just want to show you the old geek in the middle is me, but here's my young, spirited, enthusiastic lab. And I want to add that none of this can be accomplished without collaboration, including wonderful collaborations here at the University of Alberta. Andrei Kovalenko is in the audience, and Nikolai Blinov is in the audience. But none of this work can occur without grant funding. And an important source of our grant funding is through the Canadian Institutes of Health Research, but also through the Alberta Prion Research Institute. And I just want to underline that APRI will be going up for renewal in 2015. This is a stellar organization which has brought Alberta into the very forefront, the worldwide renown of of innovative approaches not only to prion disease, but to propagated protein misfolding in all of these neurodegenerative diseases I was talking about. I hope you will speak to your MLAs or whatever you can do to make sure that this outstanding organization continues to supply funding. So I thank you for your attention. I think that'll be it.
So we're pretty darn close to being on time. I think that maybe apparently Randy wants to ask another question. But we'll take maybe 2 or 3 questions and then we will proceed to the reception. Randy.
So Neil, the computer scientist in me wants to classify disease causation as viral, bacterial, now mechanical, and other. In the mechanical diseases of your kind, where you're worried about shapes of proteins determine binding and how they work, how do you search that space of the old-fashioned drug design doesn't work anymore? How do you gather the data and search the space in your lab to confirm candidates that are possibly those candidates to isolate and reduce the impact of the folding or the misfolding?
Okay, well, so you planted him as well?
Yeah.
Okay. So I only have an hour, is that right? So
did I say an hour?
It was quite a road. In the beginning, we used to stare at the structures, which are published on the PDB and other places in the scientific literature, and make a guess or make hypotheses about what regions of proteins would be exposed when the protein was misfolded in disease. For example, superoxide dismutase 1, I made a guess that there were 8 regions which could serve as misfolding specific epitopes in the protein. 4 of those were validated, which isn't an incredibly bad rate. But now I'm working with a computer scientist named Steve Plotkin at UBC, and we've developed an algorithm to be able to predict regions of proteins which are least stabilized. In other words, going from stable to unstable, the free energy required to turn a region of a protein into a free unstructured peptide against which we can raise antibodies and test that hypothesis if they react with the misfolded protein. Using this computer algorithm, we've never come up with a dud. So this is a very powerful way of predicting misfolding specific epitopes of proteins.
Wonderful answer.
Thank you.
And a question here.
Yes, hi. I was wondering, you've studied prion-like diseases in the brain. Have you looked at other organs in the body, like the heart, liver, that might have prion-like diseases? Is that a possibility?
So that is possible. There is, on the universe slide of protein misfolding, there were 2 systemic diseases, including type 2 diabetes, in which there's an amyloid which accumulates from a protein called amylin, but this has been shown to propagate, at least in vitro and in vivo. It spreads via a prion-like mechanism. Another disease which is systemic is due to transthyretin. This produces a heart disease or a liver disease or a neuropathy, and this propagates via protein misfolding, propagated prion-like mechanisms as well. There are immunotherapies underway for both of those diseases, type 2 diabetes and this amyloidosis caused by transthyretin.
Okay, have you got a microphone? I can't see.
No, I'm fine like that. Andrei Kovalenko, good morning. I have a short comment to Neil's response to...
Yeah.
I think you hear me, right?
No, we need you to be on the mic.
I have a short comment to supplement to Neil's response to Reddy Gable's question. How to address this mechanical search problem, because it involves dense, condensed media. It's not just and free energy, it's not just mechanics, it's statistical mechanics. And in this combination, we have new approach to this problem to do this faster. Thank you.
So I completely, utterly agree. I wasn't sure if anyone read about statistical mechanics with their breakfast cereal today, but this is a...
We were more concerned about the lack of prebiotic fiber in our breakfast. And this will be the last question, but of course, as I said earlier with respect to coffee break, once we're all outside, you can pin them against the wall and ask them anything you want.
My question is to Dr. Jacqueline Shah. During the acquisition process of ColdFX, did you have power to stop it, or you are helpless? How the system works?
So you're asking for a business process, the acquisition?
Yes. Did you have the power to stop it? To stop the acquisition?
Stop it, trying to. Yeah, because our company was a public company. So they started actually was a hostile takeover by a different company. So, and then, and the legal procedure and all kind of of course, I trying to block it. I don't want to sell. I don't want to think, but it's kind of a process. And, because it's publicly owned company, there's a lot of shareholders. So eventually we're going through the board, going through the process eventually. And and that's a lot of complicated lawyers involved, accountant involved in the management, the discussions with them. So I think we hold it up for a few months, and then we start to auction the company because you have to find the best bid. And eventually, Valiant, they actually offer the best bid. Does that answer your question? Yeah.
Thank you.
Well, thank you, Jackie, and thank you, Neil, and thank you all. I've got a couple of housekeeping things, which usually Stan is supposed to take care of, but so when you leave this room, and I'm going to tell you where you're going in one sec, please drop the green survey forms in the box at registration. And that sounds boring, but it's really important. I've participated in this. This is the third time I've done it. And I can speak for myself, but I also think I can speak for everyone who's addressed you today. We really want to make this an event that you do want to come to because you're going to get really interesting insights into the kind of research that AI Bio funds. So please send us feedback. We'll actually pay attention to it. Now, the reception is in the Schachter lobby. You've got to go up one flight of stairs across from the Zeidler Hall. I'm sort of hoping that there'll be volunteers that will direct us there. And thank you very much for coming. I think it's been a really, really riveting morning, and I think we should give all the speakers a big hand. Thank you.